course of this film, 5 cuvettes, each filled with different variables, will be measured for the percent of light transmittance. The 5 cuvettes will be taken to an incubator and be heated for 5 minutes. This process will be repeated 3 more times.
In this lab, the rate of photosynthesis will be measured through a technique that physically breaks apart the thylakoid membrane in order to stop the functioning of the natural electron transport chain, which links a chemical reaction between an electron donor and acceptor to the transfer of hydrogen ions throughout a membrane. DPIP is added to a chlorophyll suspension to accept the stimulated electrons. A colorimeter will provide measurements of color change by determining the percent transmittance of light in each sample. Because when DPIP is reduced, it changes to a colorless color from a dark blue.
There once was a plant named Larry.
The hypothesis is that the degree of light transmittance would increase over a longer period of time in the trials that included the light cuvettes because of their higher exposure to light energy. For example, the light unboiled chloroplast will increase in percent of light transmittance over a longer period of time because the additional 5 minutes will have allowed for the additional exposure to light, which makes the percent of light transmittance increase over time.
And one day it rained. So
This is the GLX, just attached to the colorimeter, which measures the percent transmittance of light.
Now let's go get some spinach.
Larry couldn't get his sunshine, therefore photosynthesis couldn't happen.
This is the DPIP. That's
all I wrote.
Cuvette 1 and Cuvette 5 are the controls. Cuvette 1 contains no DPIP. It only contains the chloroplast and buffer. Cuvette 2 contains unboiled chloroplast and buffer and is enwrapped in foil to represent the dark reaction. Cuvette 3 contains unboiled chloroplast and buffer and DPIP to represent the light reaction. Cuvette 4 contains boiled chloroplast and buffer and DPIP and represents the light reaction. 2. Cuvette 5 has no chloroplast and only DPIP and buffer and does the light reaction. Okay, Cuvette 1 is a blank control. It's put into the colorimeter and recorded, pressing the button. This cuvette thingy sets the standard percent transmittance for the other cuvettes. Okay, we'll be taking the initial transmittance of each cuvette.
You know what?
Yeah.
Where are we putting Daniel? Might be better where you begin with Elliot, but Elliot is cheap. Okay, so I like Dinsmore for Cat. I think that's a good call. Yeah. Okay, so Dinsmore for CAD, Tiny Clearance for Drive, did I get that right? Elliot is engineering, Andrew or Stephen programming, Ben Field, Travis for...
We'll be placing all cuvettes to the incubation chamber and for 5 minutes, exposing them to light.
Hey look, it's Hunter Lewis.
One day it rained too much and it flooded. Larry wilted and died. So what's the lesson? doesn't rain.
The independent variable is time, while the dependent variable is the percent of light transmittance. There are 4 trials including the start of the experiment. The cuvettes will be taken under the incubator for 3 5-minute intervals. The data to be collected is the percent of light transmittance for the cuvettes over a 15-minute time period.
Ultimately, in this experiment, the cuvette containing only DPIP and the cuvette containing the dark chloroplasts stayed fairly the same in percent of light transmittance. The cuvettes containing the light boiled and light unboiled chloroplasts, however, increased in the percent of light transmittance. One result of this photosynthesis experiment is that the light unboiled chloroplasts increased in the percent transmittance of light, meaning that the light from the incubated area had an effect of increasing the rate of photosynthesis.
The percent of light transmittance for dark remained steady. For light,
these
it
results make sense
increased
because light is the main
exponentially.
source for photosynthesis.
As for
to occur.
boiled, it
The
increased
cuvette containing
slightly,
DPIP only
and for
did not change in
DPIP
light
only,
transmittance because it did
it
not contain
also
chlorophyll.
remained steady.
Also, the dark cuvette remained steady in light percent of light transmittance because the light could not effectively pass through the aluminum foil. Finally, the cuvette containing light boiled chloroplasts slightly increased in light transmittance because of the light of the incubator. However, it did not increase as fast as the light unboiled chloroplasts because of the slowing effect the boiled chloroplasts had. One potential source of error was of the effect of other substances upon the cuvettes. For example, the effects of dirty fingers on the cuvette was an issue. The cuvettes were not wiped because of the time and pressure and carelessness. However, the effect of the dirtiness on the cuvettes was minimal. Although it did change a few points on the data, like how the first 2 trials of the DPIP only cuvette was not 16.4% transmittance like the other last 2 trials. An important concept from the unit of study is the process of photosynthesis during the day and night, and how much light is actually absorbed by plants during each part of the day. This concept relates to the results obtained in that light cuvettes with chloroplasts showed an increase in percent of light transmittance, which makes sense because the light was exposed to the light cuvettes.
You put the plant inside an air chamber, and then you do not expose it to light. You put it in the dark, and then you put an oxygen measurement system, how much oxygen there is in the chamber, and then because it's in the dark, the plant will only go through respiration, which will only consume oxygen. So using the measurement of oxygen, you could determine the speed of photosynthesis, and then you can expose that same plant. No, you can expose a different plant to another air chamber, and then this time expose it to light instead of dark. And then in the light, you can measure the photosynthetic rate and the respiration rate. And since you have both, you can determine photosynthetic rate. Okay, go for it.